Hello, everyone, and compliments of the new year. Welcome to our fourth Infogest Viva Webinar. We are happy to have you today. As our speaker today, we have Judit Tormási. And as panelists in the background there, we have Talita, Comunian, and Eva Hermandez, who is new to facilitating these webinars. You'll be seeing him in the next webinars. And myself, I'm Shannon, Gwala. So I'm going to introduce Judit. So Judit studies at the Department of Food Chemistry and Analytical Chemistry at the Hungarian University of Agriculture and Life Sciences. And the focus of her work was on the adaptation and application of the Infogest protocol, with special emphasis on the development of a platform for determination of triglyceride digestibility. She further investigated the impact of food containing lipase inhibitory components on triglyceride digestibility and consequently on the protein digestibility. So we will now give the floor to Judit with her pre-recorded presentation. Please do feel free to drop in your questions in the Q&A below, and Judit will be happy to answer your questions at the end of her presentation.
Welcome everyone, my name is Judit Tormási and I would like to thank the Teagasc and Infogest Viva team for giving me this opportunity to present my work, which is about the effect of foods containing lipase inhibitory bioactive substances on triglyceride hydrolysis and protein digestibility. First, I would like to introduce our little work group. We are from the Hungarian University of Agriculture and Life Sciences at the Department of Food Chemistry and Analysis. We adopted the Infogest protocol in 2019 when I started my PhD work. And since then, my supervisor and I work on many topics involving simulated digestion. These topics include lipid and protein digestibility, how co-digestion of various foods influence these mechanisms, and more recently, we started to assess starch digestibility as well. I would also like to thank here these three ladies for their help in the amino acid analysis. This presentation will focus on lipid digestibility, how it is influenced by other foods containing bioactive constituents, and also on protein digestibility. Dietary fats mostly contain triglycerides. Triglycerides consist of 3 fatty acids attached to a glycerol backbone. During human digestion, triglyceride hydrolysis is started in the stomach by the action of gastric lipase. This enzyme has a specific activity towards SN3 fatty acids, which is especially important for short-chain fatty acids, mostly found in this position. The partially digested triglycerides move to the small intestine, where the main part of lipid digestion takes place by the action of pancreatic lipase. This enzyme acts on the fatty acids in the SN1 and SN3 position and primarily prefers symmetric configuration. Symmetry in this context means that the triglycerides contains the same fatty acids in the SN1 and SN3 position. The monoglycerides and free fatty acids are taken up by different pathways and resynthesized in the enterocytes into chylomicrons, which are triglyceride-rich lipoproteins. With in vitro methods, lipid digestibility could be studied based on the monitoring of lipolysis products generated until the end of the small intestinal phase. Since the original Infogest method is a static method to fix enzyme activities and does not consider intestinal absorption, bioaccessible lipid forms like free fatty acids and monoacylglycerols at the end of the 4-hour digestion experiment could be used to monitor lipid digestibility. The mechanism is affected by the food composition, the food structure, and the co-digestion with different foods. Therefore, our first goal was to develop a method for fatty acid-specific assessment of lipid digestibility harmonized with the static infogest method, by which the effect of these factors could be easily and routinely evaluated based on fatty acid release. Also, changes in lipid digestibility caused by modifying these factors could be easily followed. During method development, there were a few questions that needed to be addressed. The first was the appropriate extraction method, which could be used for both the products and the digesta. Since the digesta is an aqueous solution containing enzymes and salts, we chose the Bly-and-Dyer method, which is based on the Falk extraction, and it uses the phase separation capability of chloroform, methanol, and water 3-component mixture to transfer lipid components from polar into non-polar medium. To test it, we choose 5 model foods and compare the fat content and fatty acid composition of the extracted foods and their digesta against corresponding isostandard methods. These test foods were baked carb, baked beef, cream, sour cream, and sour cream analog. During total fat content determination of the food products, there were no significant differences in the weight of the extracted fats. However, when we tried the gravimetric determination of total fat content at the extracted digestas, this method was not applicable. We think that this is because the evaporation or degradation of free fatty acids during heat treatment applied for drying extracted fats. For the comparison of the fatty acid composition results, we used the Blunt-Altman plots. Based on these, it can be concluded that the Bly-and-Dyer method is interchangeable with the isostandards, and there was no proportional bias between the fatty acid composition of the foods and their digesta. After the appropriate fat extraction was established, the protocol looked like this. So after the in vitro digestion, an aliquot is taken from the digesta and the lipid components are extracted with the Bly-and-Dyer method. The second question was, how can we obtain fatty acid specific analysis of the free fatty acid release? For this, we opted for isostandard methods. 2 aliquots are taken from the lower chloroform phase after the Bly-and-Dyer extraction. After solvent evaporation, one is derivatized with the so-called TFA method, which is a 2-step reaction where all lipid compounds are methylated. The other one is derivatized with the EFA method, a 1-step reaction only methylating the esterified fatty acid compounds, but not the free fatty acids. The methylated fatty acids then analyzed with GCFID. After subtracting the data gathered from the 2 aliquots, the free fatty acid content can be derived. From these results, bioaccessible fatty acid content and release ratio of each fatty acid are also calculated. In order to improve the analytical accuracy, we needed to choose an internal standard to normalize the differences between the 2 derivatization methods. We had 3 choices: fatty acid methylesters, free fatty acids, and triglycerides. Since the first 2 is not reacted in one or either of the derivatization methods, we chose an appropriate triglyceride as standard, which is reacted in both methods. This was the tri-nonodecanoic acid, since this triglyceride is composed of a fatty acid type which does not occur in any food. Finally, the last question was the selectivity of the 2 derivatization methods. To test it, we performed an experiment where besides the internal standard, heptadecanoic acid as free fatty acid and glycerol 3-heptadecanoic acid as triacylglycerides were added to the aliquots before the fat extraction. In the TFA method, both test compounds were recovered above 90%, and with the EFA method, only the triglyceride form was recovered as expected, since the free fatty acids do not react in the 1-step derivatization process. After all, it can be concluded that the developed method with the chosen internal standard is appropriate for the purposes. The final protocol looked like this, and there were 4 key advantages. The fat content of the food product and the digesta could be determined by the same method, and various steps of simple preparation were normalized with the addition of a triglyceride internal standard. The total fatty acid content and the esterified fatty acid content could be determined from the same sample, which could be used to calculate lipid digestibility based on free fatty acid release. After we established the workflow of the method, we started to get results, and all foods had relatively low digestibility. However, based on available literature data, we expected higher values. It was hypothesized that this is because of the enzyme-substrate ratio. So we started to decrease the sample size, and we saw that there was a very close correlation between the sample size and the lipid digestibility of the test food. Thus, we could conclude that the measured digestibility mainly depends on lipid content introduced to the system. So, as a methodological compromise, we suggest to use a standardized lipid content to obtain a realistic picture on the effect of an inhibitor or a treatment on the lipid digestibility. In our routine protocols, we arbitrarily set the lipid content of the food not more than 250 milligrams. Furthermore, if systematic inhibitory assays are conducted, lipid content should not vary in different experimental arms. Since our focus was on studying the changes in the lipid digestibility, we also wanted to study the effect of treatments or inhibition on the two main lipolytic enzymes separately. Therefore, we used both versions of the Infogest method. In the version 1, the pancreatic enzyme complex is used, and in version 2, in addition to these, gastric lipase is also added as a part of the rabbit gastric extract. This difference between the two versions of the Infogest methods enabled to do mechanistic evaluation of lipid digestibility. Now, I will continue with the applications of the methods. Worldwide prevalence of obesity is rising. Every second adult is overweight or obese, and overeating kills more people yearly than malnutrition. However, obesity is preventable and mainly caused by increased energy intake and lack of physical activity. Since increasing physical activity is a personal choice, the pharmaceutical industry and food industry focuses on the first cause. In Europe, the only authorized medicine to treat obesity is a drug called Orlistat. This works as a reversible inhibitor for both gastric and pancreatic lipase, inhibiting triglyceride hydrolysis. Although it is sufficient to decrease lipid digestion, it causes serious side effects which could reduce the quality of life of patients. Therefore, there is an interest in finding alternative, natural, and milder solutions for reducing digestibility of high-fat foods. A viable option could be food pairing, since there are numerous plants that contain proven lipase inhibitory agents like polyphenols or saponins. Co-consumption with these foods could be advantageous because they not only contain the active agents but additionally components which could decrease the prevalence of the side effects. In these studies, we asked two main questions. Is the same effect as shown in simplistic enzyme assays could be achieved in a rather complex digestion situation with real foods containing various forms of the substrates? And how does the inhibition affect the digestion of other macronutrients? Here, I will show our results on how foods containing lipase inhibitory bioactive substances affect triglycerides and protein digestion through co-digestion experiments with grapeseed powder and black tea brew. Grapeseed powder is a byproduct of wine production. After the juice is pressed, the remaining seeds are dried, powdered, and sold as a food supplement. This powder mostly contains tannins and condensed tannins as bioactives. Several types of extracts from grapeseed powder were tested by enzyme inhibitory assays and were proven to have pancreatic lipase inhibitory effect. First, dose dependency test was done with cream with 30% fat content as a lipid source. 5, 10, and 15% of grapeseed powder was added, and digestion experiment was obtained with both versions of the Infogest method. When we look at the control results, we see that during lipid digestion of cream, there is almost a 12% increase with the addition of gastric lipase. This surplus, however, is not evenly distributed between all fatty acids. After we analyzed the fatty acid specific data, it showed a specific increase in the release ratio of short and medium chain fatty acids. This is due to the diverse fatty acid composition of the milk fat. The triglycerides containing one or two of these short and medium chain fatty acids are asymmetric, which are not preferred by pancreatic lipase. Thus, when these triglycerides are only hydrolyzed by pancreatic lipase, the release of these fatty acids are hindered. With the addition of grapeseed powder, a 10% decrease was observed at all levels in both versions of the Infogest method. The addition of 5% grapeseed powder was also enough for the inhibitory effects to manifest, and there was no dose dependency, hence further addition of grapeseed powder did not improve the inhibitory effects. When only pancreatic lipase is added, the effect of grapeseed powder was greater on the short chain and medium chain fatty acids, so the specific hindering effect of asymmetric triglycerides are more dominant when the pancreatic lipase is inhibited. If the gastric lipase is also added to the digestion, there is no change in the pattern of fatty acid release. This shows that the inhibitory effect of grapeseed powder is only affecting the pancreatic lipase. However, due to its specificity to eccentric fatty acids, gastric lipase could compensate for some of the loss, which in milk fats are mainly short and medium chain fatty acids. After the evaluation of cream test food, we wanted to test the inhibitory effect of grapeseed powder on other matrices. So additionally, 2 co-digestions were conducted where grapeseed powder was added to baked carb and to baked beef in the same concentration as used for cream. The results show that the effect is only significant when pancreatic lipase is applied. This confirms that the grapeseed powder only affects pancreatic lipase activity. After evaluating the fatty acid specific results, the decreased release ratios also showed dominant effect on the asymmetric triglycerides. However, due to the fatty acid composition of these test foods, the inhibition of pancreatic lipase is more manifested in the reduced release of unsaturated fatty acids. Here, interestingly, we found that gastric lipase could fully substitute the loss of pancreatic lipase activity. Thus, it can be concluded that when the additional effect of gastric lipase is negligible during the initial lipid digestion of the food, the inhibition of pancreatic lipase is not sufficient to lower overall lipid digestibility. The second set of experiments are on the effect of black tea brew of the tea cultivar with high tannin content. Tea is a highly studied plant material due to its health benefits. Aqueous extract of black tea was proven as a potent inhibitory agent on pancreatic lipase. In addition, it was also showed that tea could have inhibitory effect on gastric lipase. So first, we conducted a dose dependency experiment where 1, 2, or 3 parts of tea brew was added to 1 part of cream. And this compilation was assessed with both versions of the Infogest method. The bioaccessible fatty acid content showed that the addition of 2-part tea was enough to reach the highest effect, and further addition did not result in any higher inhibition. At this level, the bioaccessible fatty acid content decreased by 22 to 25%. The fatty acid specific results were also analyzed. When only pancreatic lipase is applied, the specific effect on short and medium chain fatty acids is more pronounced by the inhibitory properties of tea than it was with the grapeseed powder. Here, the release of these fatty acids were reduced twice as much as the other fatty acids. It seems that gastric lipase has a key role in digesting asymmetric triacylglycerides containing short and medium chain fatty acids due to the gastric lipase's specificity to SN3 fatty acids. In the case of tea, this feature of gastric lipase could balance the specific inhibition of pancreatic lipase. We also tried the same experiment on another test food, namely on baked beef with the 1:2 ratio. However, no effect was observed with either Infogest method. From this, we conclude that the specific inhibitory effect of tea is important. However, these inhibitory properties are more relevant when the consumed fat contains asymmetric triglycerides, for example, with one short or medium chain fatty acids, like in the case of cream. So the take-home messages of this section are that grapeseed powder has an inhibitory effect on pancreatic lipase activity. However, if the specific effect of gastric lipase is negligible during the initial lipid digestion, this inhibition of pancreatic lipase is not sufficient to lower overall lipid digestibility. Black tea has a specific inhibitory effect on pancreatic lipase towards short and medium chain fatty acids containing asymmetric triacylglycerides, and this is more relevant when the food has diverse fatty acid composition, like in the case of cream. The other main question was how the differences or modification of lipid digestion influences digestibility of other micronutrients. So the next goal was to study co-digestion of high-fat and high-protein foods and to look into the interplay of macronutrient digestion. First, we started with lipids and proteins, since the release of these two macromolecules takes place in the same parts of the gastrointestinal tract, namely in the stomach via the action of gastric lipase and pepsin, and in the small intestine by specific enzymes secreted by pancreatin. So for this, we designed an experiment that we called the sour cream pasta study. Sour cream pasta is a traditional Hungarian dish. Additionally, we included another topping since recently there is a substitute product for sour cream called sour cream analog, where the milk fat is substituted with some type of vegetable fat. So there were three main aspects considered in this experiment. First, we looked into the difference in lipid digestibility between the two sour cream products. Then, the effect of pasta on lipid digestibility was assessed in a fatty acid specific way. And finally, the addition of high-fat toppings on protein digestibility of pasta was evaluated based on free amino group content. Additionally, microscopic analysis was used to get a clear picture on the structural changes influencing digestion. First, we compared lipid digestibility of the two toppings. Sour cream is made from cream with bacterial fermentation, so milk fat globules are in their native form covered with milk proteins. Sour cream analog is also made with bacterial fermentation. However, it is made from skim milk mixed with some type of vegetable oil, in this case with palm oil. This results in palm oil droplets covered with milk proteins. After in vitro digestion, bioaccessible fatty acid content was determined, and there was a significant difference between the two products. We also performed microscopic analysis to deeper understand the differences. The first row shows the structure of the two products, and the second row shows their gastric digesta. Here we could see that after gastric digestion, size of milk fat globules are larger than palm oil droplets. However, this difference cannot be seen between the two products. Based on literature, we think that this could be because palm oil and milk proteins form a more pH-stable emulsion, where the destabilization and aggregation in the gastric environment is less dominant than in the acidified native milk matrix. This structural change happens during the gastric digestion could partly explain the difference in the extent of lipid digestion between the two products. Next, the effect of pasta addition on lipid digestibility of the toppings was assessed. Change in lipid digestion only occurred during co-digestion of sour cream and pasta. The addition of pasta increased lipid digestibility of milk fat globules. After structural analysis, we saw that lipid droplets are attached to the surface of pasta proteins. Therefore, pasta protein acts as a carrier for milk fat globules, and by this means perhaps creating a better steric accessibility for lipases, which increases the effectiveness of lipid digestibility. Difference between released fatty acids were also evaluated after sour cream digestion, without, and with pasta. We also found a significant increase in short and medium-chain fatty acids, which are associated with asymmetric triglycerides and gastric lipase specificity. This confirms that the effect that modified lipid digestibility of milk fat globules happens during gastric digestion. Next, we looked at the protein digestibility of pasta and its potential modification caused by the high-fat toppings. To study protein digestibility, we used the method presented by Raquel Sousa last month with slight modifications. After in vitro digestion, aliquots were taken from the digesta, and the undigested protein fraction was isolated with methanolic precipitation. This methanolic extract was further hydrolyzed in hydrochloric acid to break down remaining peptide chains. Evaluation of protein digestibility was based on free amino group content with the OPA method. This table shows the unhydrolyzed and hydrolyzed OPA results. During lipid evaluation, we concluded that the addition of pasta proteins increased lipid digestibility of milk fat because these proteins serve as a carrier for milk fat globules. This effect causes a slight decrease in the free amino group content, so the protein digestion seems to be hindered due to it. Although it does not have a major effect on overall protein digestibility. Based on these results, we hypothesized that different types of fats could have distinct effect on protein digestibility, so we conducted a series of experiments where pasta was co-digested with 11 types of vegetable oils. The protein digestibility results from the methanolic extract before and after hydrolysis are shown on this graph. Since we wanted to know the systematic effect of lipid composition on protein digestibility, several lipid composition indicators was created and correlated with the measured protein digestibility. Our first results showed a high correlation with oleic acid content. However, at this point, to double-check the validity of this observation, amino acid-based protein digestibility was also measured, since our platform has improved during this period. After evaluating this data, we saw that on the contrary to the results obtained with the OPA method, there was no correlation with any of the indicators, so there was no systematic effect of fatty acid composition of the added oil on the protein digestibility of pasta proteins. Errors in analysis not only occurred during this experiment. Unfortunately, when we wanted to evaluate the effect of bioactive containing foods on protein digestibility of baked carp and baked beef test foods from the OPA results after hydrolysis, practically it could not be performed. We think that this is because of the high fat content of the samples. Based on literature, we also think that this is because of the complex forming capability of oleic acid with peptides on high temperatures, and this affects both the acidic hydrolysis and OPA reaction. So we concluded that the standard OPA method that we use is not appropriate for the high-fat matrices. Thus, we have started to work on an improvement of the sample pre-treatment protocol of OPA method, including a defatting step. Hopefully, soon it will be ready and we will be able to present new realistic results on the interplay of lipid and protein digestion, which rely on solid analytical data. So to conclude this section, we found that the addition of pasta proteins increased the lipid digestibility of milk fat globules, and based on structural organization and fatty acid release, this effect is expressed in the stomach and consequently results in modified protein digestion. We also could conclude that different types of oils have effect on the protein digestibility. However, this effect is not systematic with the fatty acid composition. We also could conclude that the high fat content, especially the high oleic acid content, could influence the OPA analysis in a systematic way. Thank you for your attention, and I am happy to answer any questions that you have. Welcome.
Thank you, Judit, for your great presentation. It was really nice. It was very interesting. Now we are going to have some questions. We have a couple of questions here. So the first one is, according to your claim, the extent of the beneficial effect, like the usefulness of the functional food containing lipase in inhibitory bioactives, depends on the type of the substrate, is practically on the type of triglycerides being digested?
Yes, hi everyone. Thank you for the question. Yes, so as the examples showed, we saw that when the food source doesn't have varied fatty acid composition, then the effect could be very negligible. So we think that the inhibitory effect of each bioactive containing foods will depend on the fatty acid composition and of the lipid source. So this could be a very interesting thing in the future when developing functional foods in this way.
I see.
Okay. Hello, Judith. Thank you for the nice presentation. Congratulations. And we have another question for you. Why did you choose baked carb, baked beef, and cream as the test food?
Yes, thank you for the question. So we choose these test foods based on our purposes on the evaluation. So we choose for cream, for example, because it's mainly containing lipids. So it has a 30% fat content and negligible carb and protein content, basically. So it is a good lipid source for evaluating only lipid digestibility. And we chose the others like baked carb and baked beef because they contain protein and fat in a very high amount. So we can evaluate the interplay between these two macronutrients, basically.
Thank you.
So the next one, we have a couple of questions here. It's good. The next one is, what do you think that could be the reason that different fats exert different effects on the digestion of pasta proteins, if not their fat acid composition?
Yeah, so we try to look into some type of surface properties when we had this question. So we evaluated surface tension and the wetting properties because we thought that this could be some reason for it, but we found no differences between the 11 types of oils that we studied. So for now, we don't really know, but we are looking into other type of surface changes that can happen in the gastric environment. Yeah.
Thank you very much.
Okay. And another one is, you say that the OPA method is not suitable for determining free amino acid groups from the digesta of high-fat foods. What modification of the method or what alternative method would you suggest to solve this problem?
Yes, it's a good question. So as I mentioned, we are trying to do another sample pretreatment method, including defatting into it. And based on this, we can try to make an integrated method, both good for lipid and protein digestion evaluation. So after the, like in one step is extraction at the step. Yeah.
Thank you.
Also, another one from Wiki. Hi, thank you for the great presentation. Could you give any recommendation and elaborate more on the enzyme lipase activity to substrate amount of lipid ratio in studying the inhibition of lipolysis?
Yes, so when we first started to get results based on our method, we show like very low lipid digestion, lipid digestibilities. So we think that the substrate ratio is too high to get an efficient enzyme activity because of the overload of the enzymes. So that's why we started to decrease the sample size, thus the lipid content of the included lipid sources. So yeah, we think that is it's like a methodological thing. So yeah.
Perfect. So we have more, yeah.
Okay.
We have a couple. Good.
Yeah.
People are interested.
Okay. Can we get some background information about the grapeseed powder and black tea used in the study of the effect of their bioactives?
Yes. So the grapeseed powder that we used is a commercially available product manufactured in a Hungarian winery called Bock Winery. They are selling these powdered grape seeds in like a high polyphenol food supplement way. So that's the one that we used. And the other one, the teas are from a local Budapest shop. And we choose this specific cultivar based on its high tannin content, basically.
Okay, thank you.
Thank you. Another one, the pasta also contains a lot of starch. Did this not interfere in the microscopic images?
Actually, no, thank you for the question. It did not interfere, but we can saw the starch granules in light microscopy as well. So, but we also did dyeing of the lipids and the proteins, so we could separate the starch during the microscopic analysis.
Perfect. Yeah.
And we have one more question for you. And this question is from Anne Mainier. You mentioned a pancreatin activity of 100 units per milliliter. If it is well recalculated,
I think so. Yeah.
to lipase, it was very low, 2000 units per milliliter in Infogest 2.0. Is that a typo?
Yeah, maybe. Sorry for that. I will check it.
Okay.
Yeah, because we use the Infogest protocol as is, so it must be.
Okay, thank you.
So we have more. We still have time, yeah? We still have a little bit of time. So for the next one, what prompted you to study the effect of tea bioactives on beef fat digestion? Is there a reality that someone eats beef with tea?
Yes, it's a highly unusual combination of foods. It's not the choice of beverage for beef. So we basically chose these test foods, as I mentioned, to study the lipid and protein digestion simultaneously. So that's why we chose to add the tea to it. So it's more of a the experimental design choice than a realistic food conception choice.
Great. We have another question or maybe comment from any Manier.
I think it's the continuation of the other.
Yeah, the continuation of the previous, the last question. So it was the activity related to trypsin.
Which type of activity? Sorry.
The question about the pancreatin activity.
Yeah.
Oh, yeah.
Yeah. So, and the next one more question. I think we can do one more. Yeah. So you have noticed that the extent of lipolysis depends on the size of the digested sample, more precisely on its fat content. Couldn't this have caused a problem during the study of the effect of bioactivities on fat digestion? Could you have taken this into account in some way?
Yes, it could have, but we designed these experiments based on the lipid content. So we chose, like I mentioned, the 250 milligrams was our limit. So we chose a lipid content under that. And we set the ratio of the bioactive containing foods to it. And then we continued with the same ratio between experimentals. Between experiments. So we use the same lipid and bioactive ratio always during these experiments.
We have one more. Do you want to finish this last one? And then we, yeah.
Sure. So the last question. Has the inhibitory effect of orlistat on fat digestion been studied on the platform you use? If so, what results were obtained and how do they compare to the magnitude of the effect exerted by natural bioactives?
Can you repeat the first part of the question, sir?
Yes. Has the inhibitory effect of orlistat on fat digestion been studied on the platform you use?
Yes, we use the orlistat as a positive control during method development and during the experiments. And yeah, compared to the natural inhibitors, it had a higher, way higher effect. So the inhibition was almost complete with the orlistat addition. Around like 2% was the lipolysis extent. So yes, it's working on the platform, and and it's much more potent inhibitor than the natural ones, as expected.
Okay.
Thank you very much. I think we don't have time for questions anymore, but thank you for your time. It was a really great presentation. It was very interesting. We can send to you the other questions we also have here. Yeah.
Yeah. Thank you.
Thank you. And now I'm going to share one slide for one announcement. So our next speaker will be Muireann in February. She's from Wageningen. In March, we have Elise from France. And in April, we have Marcus from Agroscope. We still have a spot available in May. So if someone is interested to present, please contact us here. And for June, we also have Jessica from Belgium. Okay, so thank you everybody for being here, and see you in the next webinar in February. Yeah, thank you.